Guys, we are now in the very last little piece of the cell lecture. We're going to be talking about cell cycle, which is basically just a way to think of like life cycle of a cell. So most of a cell's life cycle is spent in what's called interphase. Interphase. Inter means between. And so interphase is the non-dividing phase, which means that they're not going to be doing mitosis. They're just hanging out doing what normal cell stuff do. Okay, so they're just being their normal cell selves. The one thing that is important to note, though, is that during interphase, that is also where you're going to be replicating the DNA. Other than that, though, they're just doing their normal function and growing. Only about 10% of their time is going to be spent in mitosis. And mitosis is the division of the nucleus, making new cells, also known as cell division. So let's talk a little bit about terminology. So here would be the cell in interphase. Once it goes into mitosis, we call that the very first kind of original cell is called the maternal cell. And the reason we say that is because we say that the maternal cell creates daughter cells. So daughter cells just means two resulting cells that came from one. One is the maternal cells, two cells that come out of the other end, those are called daughter cells. There's nothing kind of different about them, that's just what they're called. So those daughter cells are going to be clones of the original maternal cell. Do you know what a clone is? Clone means they're genetically identical. They have the same DNA, and not only do they have the same DNA, they're also going to have the same expression patterns. And the reason that's important is because if you are a heart cell, you better be making heart cells at the other end, right? So you want to create cells that will have the same function as the original maternal cell. Okay, so as we go through the stages of mitosis, for each of the phase that I'm going to talk about, you need to be able to recognize it by sight. So if I show you a picture of it, you should be able to know what phase it is. That's going to be important for lab as well. You also should be able to recognize by description. So that means you need to know what happens in each of those phases. So when we go through mitosis, there are 4 phases. So we have prophase, metaphase, anaphase, and telophase. The way I remember that is peanut. That's just how I remember it. It's not an official term or anything. So prophase, metaphase, anaphase, and telophase. So we're going to start in prophase. Now, if we looked at this picture right here, this cell is in interphase. And one thing that is distinctive about interphase is that you see a very clear, distinct boundary for the nucleus. The nuclear envelope is fully intact. This cell here, this cell is now in prophase. And you can see that instead of having a clear, crisp boundary, clear, crisp nuclear envelope, now you don't really see where the edge of the nucleus is, and that's because the nuclear envelope is gone. It's just moved apart. And the reason for that is because the function of mitosis is to take that genetic material that you've already replicated in interphase and move it to the poles so that you can create 2 cells from it. So you can't move things if they're contained. So we got to get rid of the nuclear envelope. So if you're looking through the microscope, this is what it will look like. It'll have clumpy DNA with no nuclear envelope. The reason the DNA is clumpy is because you're going to be moving these long strands of DNA around. You're not just going to ship them as is. You need to coil them all up into a nice, neat little package. So I like to think of it like Christmas lights, right? So if you've got Christmas lights and they're all spread out, if you were to just like start moving around with those Christmas lights, they're going to get all tangled. They might get damaged. So you need to bundle them all up, package them up. That way you can ship them easily. Okay, so you're going to coil the DNA So you'll see that clumpy DNA here, and you're going to get rid of the nuclear envelope. That's going to allow you to be able to move it, okay? The third thing that happens is the formation of what's called the spindle apparatus. Okay, so these structures right here, those are called centrioles. And those centrioles will start creating a cytoskeleton structure, create those microtubule structures that will come out. They kind of look like little stars. And what they do is they create a framework that will attach to the chromosomes and allow them to be pulled. Okay, so remember, cytoskeleton helps move things around. Now we need a specialized cytoskeleton in order to move the chromosomes around. That's called the spindle apparatus. Okay, so the next phase of PMET is metaphase. So M is for middle. M is for middle. So if we look at this cell right here, this dark thing right here, those are the chromosomes, and you'll notice that the chromosomes are all lined up along the middle. That's called the metaphasic plate, right along the very, very center here. These star structures, that would be the fully intact spindle apparatus that was formed starting in the prophase. Okay, so the major event of the metaphase is that the chromosomes line up in the middle along the metaphasic plate. The next one would be anaphase. So during anaphase, the chromosomes that are all lined up, now they're going to be pulled apart from one another. Okay, so we had 2 copies of the chromosomes all lined up, ready to go. Now they're going to be pulled apart from one another. And so when you look through the microscope, the major difference between the metaphase and the anaphase is that instead of having 1 line of chromosomes, now you have 2 lines of chromosomes with some space in the middle. If you have 2 lines of chromosomes, you have to be in anaphase because the major function of anaphase is to pull those chromosomes out towards the poles or towards the edges of the cells. Okay, so any space between them at all, now you're in anaphase. Okay, last one, telo. Telo means in, so telophase is the last phase. And for the telophase, it's basically, in terms of the events that happen, it's basically just the opposite of what you did in prophase, which is good news because if you know what happens in prophase, you also know what happens in telophase. It's basically just undoes everything that it already did. So the first thing is those chromosomes are going to uncoil. So instead of be packaging nice and tight in chromosomes, they're going to uncoil into what's called chromatin. And then the other thing is the spindle apparatus. Don't need that anymore. That'll go away. And then you're going to start reforming that nucleus. Now, all of that would be part of telophase, but in fact, the most visually kind of distinctive part of telophase is the presence of another process that's happening at the same time. That is called cytokinesis. Cytokinesis. So technically, mitosis is the division of the genetic material. Cytokinesis is the division of all of the rest of the cell. Okay, so for animal cells, the way that it works is if you have one big ball, I like to think of like a ball of dough. The way that you can take that one big ball of dough and make them into 2 is by pinching them off in the middle. Okay, so what happens here is you have this contractile ring. It's made up of proteins, little protein contractile ring, and it slowly tightens that contractile ring. And what it does is it buds them off of each other by just sort of squeezing in the middle, and then 1 will become 2. Okay, so the cytokinesis is going to begin during telophase. Okay, so you're going to start the squeezing in the middle part. And because of that, you're going to have the presence of this little indentation that's called the cleavage furrow, Okay, so if you see the presence of the cleavage furrow at all, that must mean that cytokinesis is occurring, and therefore you know that that is in telophase. Okay, So anaphase, 2 clumps, but no cleavage furrow. See, nice and smooth there. Now we're in telophase because I see the presence of cytokinesis happening at the same time. Okay. So that's the end of it. I just wanted to remind you that you do have your very first lecture quiz next week. For the quizzes, they have to be done on a particular date. It will be available for 24 hours on September 9th. So make sure you take it on that day. You won't be able to take it any other day, but you can take it anytime that you want to during that day. These will be worth 15 points in the quizzes categories, so they are heftier by far than the mini quizzes. Mini quizzes are like practice. This one, make sure you study because you can only take it once. Okay, and it's going to cover everything from week 1, which means the introduction to anatomy. Those are the things like, you know, the directional terms and all of that, and the planes and the body cavities, and then the cellular structures and the cell cycle that we just talked about. Okay, bye-bye.